Our next speaker is Mr. Ren Marquez, who is a PhD candidate in Caltech. Ren, are you there? Okay. You want to share your slides?
Let's see here.
Try mo. Sir Rene Riesa is also in our Zoom meeting, one of our distinguished alums. And Miko, by the way, before I forget, merong mga message sa'yo si Dr. Yumul about the geochem of your study.
Opo.
Okay, take it away, Ren.
Okay, thanks, sir. Can everybody hear me okay?
Yeah, we can hear you.
Okay, sorry about the mask. I'm still on campus and I'm basically required to wear it. So yeah, so this is a work that I did actually more than a year ago now with Dr. Jess Adkins, basically trying to understand deep sea microbial sulfate reduction and how that actually relates to atmospheric oxygen and basically the global redox cycle. Okay, go on, there we go. Right, so the basic task that we're trying to accomplish here is just to, well, describe sulfur cycling, specifically in deep ocean environments. And before we try to dive into that, let's first just take a second and like have everyone on the same page and understand that there is this very intimate coupling between carbon, sulfur, and oxygen and their respective cycles in trying to control how much oxygen is actually in the atmosphere. So everybody has seen this type of reaction before. You take carbon dioxide and water, and you can convert that into glucose, basically biomass, and it has oxygen as a byproduct. So this is photosynthesis. The reverse is, of course, respiration or remineralization. And something that's not really talked about as much is that in geologic timescales, if there is an equal amount of photosynthesis and respiration going on, there's actually no net production of oxygen, right? Because you're just, all of the product you're making, you're just taking it back, nothing's gonna happen. But if for some reason you take some of this organic carbon and just take it away somewhere, bury it somewhere, there is going to be an equal amount of oxygen that's not being used, and that could actually make its way into the atmosphere. And so that kind of process and that kind of coupling happens in geologic timescales, so hundreds and hundreds of millions of years. Right? But of course, oxygen is not the only thing that's available for organic carbon respiration or remineralization. There are other oxidants out there. We have nitrate, we have manganese and iron oxides, and we have sulfate. And so you can actually oxidize organic carbon with sulfate in a process known as microbial sulfate reduction. And the way that this goes is that if there's oxygen available, it's going to be utilized. The aerobic respirators are just going to dominate because there is a lot of energy to be derived from aerobic respiration. And once it runs out, you go to nitrate, you go to manganese oxide, you go to iron oxide, and then to sulfate. And if that is the case, why do we actually care about sulfate reduction? If there's not a lot of energy to be derived from it, why do we actually care? Well, for one, there's a lot of sulfate going around in the ocean, right? It's about 28 millimolar, compare that to manganese and iron, about micromolar to nanomolar scale. So there is a lot of sulfate going around, that there is a lot of sulfate reduction that could go on. And so there is this very important thing that we have to consider when we're trying to account for everything that affects atmospheric oxygen. Again, we could have just burial of organic carbon, and you could actually just use something else to oxidize organic carbon. Just don't use oxygen, use sulfate instead. So this is a very simplified version of what goes on during sulfate reduction. You take 8 moles of sulfate, and you can actually oxidize about 15 moles of organic carbon. And the resulting product is, of course, sulfide in the form, or at least stored in the form of pyrite or iron sulfide. And so when we're trying to account and model all of these things, we have to keep track of first organic carbon, of course, and then iron sulfide, the main byproduct of sulfate reduction. And indeed, for decades, people have actually tried to do this whole mass balance thing, right? So when doing a mass balance, you just take whatever comes in, must come out, assuming that there is some sort of steady state, which, you know, in geologic timescales is a fair assumption. And the main outputs are, of course, the formation of evaporites, which are mainly barite and some gypsum in the early Earth. And so that has a certain isotope composition, typically reflective of seawater. And another output is, of course, the formation of pyrite through sulfate reduction. And there is an extra term here. As you can see, that is the cap delta 34S. So it's basically the offset between the resulting sulfide being sequestered in the pyrite and the starting sulfur isotope composition of the sulfate. And indeed, when you look at the record for the past 4 billion years, there is some sort of constant offset between the sulfate reflective, again, of the seawater composition and that of the buried pyrite. There is some sort of negative offset here. So in the y-axis, you just have the sulfur isotope composition. And that makes a lot of sense if you look at the bacterial culture data, because when we actually try to culture these sulfate reducers, what we find is that there is, again, a constant negative offset between the resulting sulfide with respect to the starting sulfate composition. And what we see from these experiments as well is that it actually pretty much matches what we see as from ab initio calculations of the theoretical limit for dissimilatory sulfate reduction, basically what happens during microbial sulfate reduction. But that offset, that epsilon, it's denoted as epsilon here. And it kind of varies in the literature, but anyway. Basically what we're seeing here is that it's not always constant, and it's actually highly dependent on the sulfate reduction rate. How fast are you sequestering the sulfate and converting it into sulfide? We see that for a very high sulfate reduction rate, it's the expression of this sulfur isotope fractionation is not really that evident. It's about 20 per mil. But if you go to a very, very slow sulfate reduction rate, it starts to approach that theoretical limit, about -70 per mil. Now, the next question is what actually controls the sulfate reduction rate? We know that it controls the epsilon, but what controls the sulfate reduction rate itself? Well, when we try and look into the global distribution of sulfate reduction, in the global ocean, we see that the highest rates are typically found in coastal environments, which makes a lot of sense because these are sites of a lot of upwelling. There's a lot of primary production going on, and there, in turn, is a lot of organic carbon rain going down. And so just the availability of the substrate, the thing that's being oxidized, actually controls sulfate reduction rates. There are other things out there that could control the sulfate reduction rate, but to the first order, this is what controls sulfate reduction. And so given this distribution of sulfate reduction in the modern ocean, most of the literature, at least in the past 4 or 5 decades, have actually focused on looking at shelf environments, just because, well, for one, the thinking is that most of the sulfate reduction is going on here. And so if we're just trying to account for sulfur cycle globally, maybe just focus on that spot. And another thing that is very crucial when trying to understand these or trying to measure these sulfur isotope compositions is the amount of sulfur to be measured. So there's a lot of pyrite being buried in this site. So in terms of instrumentation, it's much, much easier. Okay, it's not going, oh, there you go. But there is one problem, right? The site-specific sulfate reduction rate is indeed higher in these shelf environments, but the problem is the aerial extent is not that high, at least compared to the continental rise and the abyss. And so that flux term that you saw from before, that's not just the specific rate in one particular site, it's integrated across the whole area. And so when you try to account for that, maybe there is some sort of leverage coming from the deep ocean. And that is what we're trying to get at here. Is the deep ocean going to influence our calculations, or at least the most accepted calculations for trying to account the global sulfur cycle, especially when trying to relate it back into atmospheric oxygen? Okay, so aside from the thing that I started with before of just trying to describe sulfur cycling in deep ocean environments, we're trying to also ask how does this actually play into the global sulfur budget? Okay, so in order to try and answer this question, we turned into one particular site, just because we have the course in the freezer upstairs. This is Site E1474 and Expedition 361 from IUDP. And I would be the first one to point out that doesn't seem like a deep ocean site. Well, in terms of the depth, for our characterization, it is, but we chose it because, for one, the pore water profile, which you're gonna see later on, is actually pretty much one of the most important things for the interpretation of this data, is relatively simple. And since we're trying to develop a new method here, maybe just start out simple and then move on to the open ocean sites later on. Okay, so come on. Right, not important. Okay, so I'm also not going to discuss the data that much, but Basically, we developed a new method using the EAIRMS. So it's an elemental analyzer. You can actually just take in your sediment, combust it, and runs through a column, and you get your sulfurized composition. The main challenge was to actually try and do it with a very low sulfur load. So very low concentrations, or at least we expect them to be very low concentrations, given that these are deep ocean sites. And so that is the main challenge here. And we were pretty successful in the calibration here, as you can see. So what did we actually see from the sites? So this is the sulfur isotope profile for the sediments in this IODP 1474. In the y-axis, we have the depth. We have two x-axes here. We have the open red circles is the weight % sulfur, which we thought at first was just pyrite. You're going to see later on that that's not the case. And then we have the blue squares over here representing the sulfur isotope composition of that sediment. And one thing that is kind of scary here is that when you try and plot the reciprocal of the weight fraction of the sulfur in these sediments versus the amount or the sulfur isotope composition, it plots as a line, which is it a Rayleigh distillation kind of thing? Is it a sort of mixing relationship? We don't know. In order to try and address the latter, we just took some of these sediments, looked at them under the SEM, and basically this is what we found. Well, no surprise, we found pyrite. And something that's very interesting here is that you see this very globular shape for the pyrite with these rhomboids. So the current accepted knowledge is that this sort of morphology is associated with microbial mats. So that's pretty good. We're pretty much sure that this pyrite is actually coming from sulfate production, not some sort of hydrothermal activity or whatever. And then there's barite. So for those of you who are not familiar, barite is a barium sulfate. And that is a problem because if there's two things that are carrying sulfur here, one is representative of sulfate reduction, the other is not, and we're not really interpreting the right data. So we actually have to correct for the presence of barite, which is not easy because it's kind of hard to dissolve barite. It takes a few months or a few weeks. And we're actually still sort of kind of testing this method at the moment. But in the meantime, one could actually just assume that, okay, barite probably has the same sulfur isotope composition as seawater, and you can actually correct for it. And then once we actually figure out the methods, let's compare what we're seeing here. But if we just do that correction now, we can actually see here that the resulting profile is relatively straight, right? And that is not really super surprising because we see this a lot in coastal environments as well. And so the average value for the profile is about -43.8 per mil. And when you compare that to the 21 per mil isotope value for seawater, that's about an epsilon value of -65 per mil. Very, very close to the thermodynamic limit, right? And so it kind of tells us now, very, very early, that the sulfate reduction rates in these sites are very, very slow. And our theory as to why this is actually straight is because pyrite formation is likely restricted to shallower zones. We could actually support that when we look at the profile for iron, because we see that it's relatively quickly depleted in the upper parts of the pore water profile over here, which I don't really have the time to discuss right now, but perhaps the more important framework to look at this data around is by looking at the pore water profiles as well. So basically what we did was just take some of the pore water and then look at the sulfate that is surrounding these sediments, just take the sulfur isotope composition. And this is a relatively simple approach that's been around since the 80s, but nobody seems to actually understand what's going on. But it's relatively simple. So just as a brief overview for everyone. So if you assume steady state, so there's no change in concentration over time, so it's just zero. The pore water profile that you're seeing is basically controlled by diffusion, advection, and something else, right? And that something else we know is sulfate reduction, at least for the most part, it's sulfate reduction, right? For the relatively low sedimentation rates that we have here, you can basically remove advection. So that diffusion term and the sulfate reduction should actually just cancel each other out. So this is a very, very good example of where the physical features that you're seeing, the physical and chemical features that you're seeing is actually a result directly, or at least could be equivalent to biology, which is very, very exciting. And you can just calculate that sulfate reduction rate. which resulted, at least in our site, as 0.65 micromoles per square centimeter per year. And when you actually compare that to the modeling data, so the ones that I showed you from before are modeling estimates for sulfate reduction rates, it actually fits pretty well for the depth range that we're looking at, which is, it's always comforting when you see that modeling actually matches with the real world. Not always the case, or maybe like 90% never the case. But another very interesting calculation that we could make here is just assume some sort of cell abundance in these sites, which, it's not as well constrained, but there is data out there. And we can have a very much a first order estimate of what the cell-specific sulfate reduction rate is. And for site U1474, it falls over here. And that is actually much, much lower compared to what we're doing in cultures. So it raises the concern of like, okay, maybe these bacterial cultures that we're doing are not really reflective of what is actually going on in the ocean. And another thing that we notice here is that even though it's so far away from what we're doing in bacterial cultures, it still matches the thermodynamic limit that we're seeing from ab initio calculations. So that is very, very comforting in the sense that, okay, we actually know something about what's going on with sulfate reduction. But I think the most important takeaway from here is that, okay, now we know that the sulfur isotope composition that's coming from sulfate reduction in the deep ocean is very, very low. And that gives us another knob to tweak, right? We know that the fluxes typically are higher in shelf environments, but the offset is much higher in deep ocean environments. So you can do this back of the envelope calculation of how much each type of environment, ocean environment, could actually shift the global ocean sulfur cycle. And so that modeling project is underway, is actually about to be published soon. But yeah, it just tells us that, okay, there is still a lot of like imagine 4 decades of data just focusing on one specific type of environment, and then just seeing this data, okay, there is actually more to this than we thought, which is, kind of alarming, but also pretty exciting for a young scientist, I think. It tells you that whatever is an established, knowledge or convention is not exactly, as strong as you would think it is. All right. Okay, so just to summarize everything that I talked about, we saw that pyrite formation and burial in IODP sites, typically restricted in the shallower parts of the sediment profile. Sulfur reduction rates are relatively slow, and so this results into a very low sulfur isotope composition, and thus has a huge swing in terms of trying to determine this mass balance between the different fluxes of sulfur and how that relates into the global redox cycle. All right, cool. Thank you so much.
All right, thanks, Ren. Of course, you didn't touch on the other implications of studying the sulfate as a source of energy. No, of course, we go back to the time when there's limited oxygen in the environment, and potentially all the microbes at that time were using other forms of energy, particularly sulfate reduction. Diba? So it's good that people are looking at this system once again, even though it's not anymore the dominant source of energy for life here on Earth.
Oh yeah, for sure.
Yeah. Are you continuing this for your PhD?
Unfortunately, I actually stopped working on this over a year ago.
Okay.
I'm focusing on 4 other projects right now, so I had to drop one.
All right.
Yeah.
Great. Thanks, Ren.